Hello, my name is Alex Brace. I'm a computer science graduate student at the University of Chicago, advised by Arvind Ramanathan and Ian Foster. Today I'd like to tell you about a project we've been working on where we are applying deep learning to an important task in computational biology called homology detection. Specifically, I'd like to focus on how the combination of domain-specific foundation models and high-performance computing can enable science at greater scales than is currently possible with traditional approaches. Before diving into computational biology, I'd like to start by giving a broad definition of the foundation model paradigm and how it has impacted science. A foundation model is originally defined as any model that is trained on a broad data collection, generally using self-supervision at scale, that can be adapted via fine-tuning to a wide variety of downstream tasks. The power of this approach is twofold. First, through pre-training larger and larger neural networks, features that implicitly capture aspects of the data emerge automatically. And second, the homogenization of the underlying learning algorithms, scalable model architectures, and even the model itself has fueled the application of FM-based approaches in computational science. Foundation models are actively being developed across a wide variety of science domains, and there's now even a dedicated SciFM conference, which will be hosted in Chicago next year. In my own research area of computational biology, a new wave of approaches are actively being developed, which provide a new lens on evolution, as well as a path to designing biological molecules capable of carrying out new functions. So before diving into the details, first I'd like to give a bit of background on this application. Proteins are large molecules composed of a sequence of amino acids. That sequence determines the resulting three-dimensional atomic structure of the protein that ultimately allows it to function in a biological context. The application we'll discuss today is referred to as homology detection, which in our context is the task of identifying similar sequences as a proxy to infer a shared evolutionary ancestry. This idea dates back to the 1950s and continues to be an active area of research important to protein function annotation, predicting the 3D structure, as with AlphaFold, as well as designing new proteins. However, today's world is much different. Advances in genome sequencing have outpaced the development of new algorithms for analyzing sequences, so new tools are needed to keep pace with the exponential growth in biological data. Fortunately, biological foundation models show promise for overcoming some of the computational barriers presented by traditional tools. Specifically, protein language models are large neural networks trained over big collections of protein sequences. These models learn to represent sequences in an embedding space, which effectively acts as a coordinate system for the protein space, allowing comparison and exploration of evolutionary relationships. So in this work, we'll ask the question of whether protein language models can effectively scale homology detection applications to large database sizes of hundreds of millions to billions of sequences. We'll explore the use of binary quantization to speed up sequence comparison. We'll then show model scaling relationships on a benchmark set. And finally, we'll investigate approximate search strategies and demonstrate the performance. The main computational component of our approach relies on vector databases, like VICE, to accelerate the search component. Vector databases provide a way to store and efficiently retrieve from large collections of vector embeddings. To build a vector database, first a large collection of protein sequences must be embedded and stored alongside a sequence identifier. At retrieval time, a sequence is embedded with the same model, and the nearest neighbor search identifies the top set of similar proteins within the database. So there are many ways to compute the k-nearest neighbor set. The cosine similarity is probably the most common method across a wide range of NLP and science applications. However, it requires the use of higher numerical precision. To make search more scalable, we explore the use of U-binary compression and Hamming distance to compute the neighbor sets. U-binary compression simply works by converting any positive vector embedding components to 1 and any negative values to 0. This results in bit vectors, which are compared by counting the number of bits that differ, which is very efficient on a CPU. And what is very surprising is that binary compression is actually more accurate than cosine similarity for large models across several diverse benchmarks we constructed. This trend is contrary to findings in NLP, and we're still trying to understand the cause of this. But we see that for large models, the accuracy approaches the gold standard traditional methods like BLAST. Now that we've seen the accuracy of the approach, I'll walk through our approach to scalable search. For this, our target search database is the UniProtKB knowledge base, which contains roughly 252 million protein sequences from across life, which result in about 1.3 terabytes of embeddings. The first phase is precomputing embeddings. For this, we make use of Parsl to distribute our workload over roughly 128 nodes of the Polaris supercomputer at ALCF. In general, this embedding computation is very similar across many foundation models from different domains. The job characteristics usually require a single GPU per model instance, for which input data is distributed in an embarrassingly parallel way, as each input sequence represents an independent unit of work. We make use of several optimizations, including an object registry, which keeps models warm in GPU memory of the Parsl process between task invocations. Putting this all together, we were able to embed 252 million sequences in roughly 3 hours, which gives us an overall throughput capable of processing 1 billion sequences in less than a day within a single job. Once embeddings are computed, the next phase is search. In order to scale to billions of sequences, approximate search strategies must be used to achieve a reasonable time to solution. The strategy used here is referred to as an inverted file index, which first partitions the entire protein space into a set of bins, each represented by a centroid. The query vector is first compared to each centroid using a Hamming distance calculation, and then exhaustive search is performed within the top clusters. As the number of clusters increases, more of the space is searched, providing a better approximation to an exact search algorithm. As it turns out, the time complexity of this algorithm scales sublinearly by the square root of the number of entries in the target database. We benchmarked the FICE implementation on a synthetic dataset out to 1 billion vectors and find that for single query search, the set of candidate hits can be reliably obtained in 1 second. With this, we can search the full UniProtKB in under 1/10 of a second. As for the space complexity, inverted file indices scale linearly with respect to the number of embeddings. In the case of UniProtKB, our in-memory storage footprint is 80 gigabytes, which easily fits into the host memory on a standard compute node. To understand the accuracy trade-off of IVF search versus exact search, we built a benchmark to compare the predicted hits of the two approaches. We see that we need to search 16 clusters out of the roughly 16,000 total clusters in order to achieve greater than 95% accuracy. Putting this together with the scaling results, this means that we can accurately search UniProtKB for full genomes in under 20 seconds. However, this is only the search time and does not include the cost of computing query embedding. So when we add in the query embedding computation, we see that for single sequence queries, the application runs in about 15 seconds, whereas for full genome, we get results in about a minute. For single sequence, the embedding largely dominates the runtime, and we are working on several optimizations to improve this. Comparing this to the newly released optimized version of MMSeq 2, we see that single sequence takes about 4 minutes and full genome takes up to 8 hours, making our approach competitive for large databases. So we've seen the scalability of this approach, and now I'd like to share one of the ways in which we are using it. As part of a much larger effort, we are hosting this application on a dedicated server to interface with the Department of Energy Systems Biology Knowledge Base, or KBase. KBase is a community research platform that helps address a wide range of biological problems. As part of it, we're using our search service to connect user-specific protein sequence data that they may have sampled in the field to the broader ecosystem of data within KBase, which includes protein structure, functional information, literature, and more. KBase is undergoing upgrades to integrate an agentic layer, allowing multiple data sources and foundation models to interface with each other, for which our tool serves to connect these various modalities to ultimately help users navigate the protein space and address questions in functional genomics. So what are the takeaways? Homology detection at scale requires faster and more scalable tools that integrate seamlessly into agentic workflows to avoid bottlenecks. Our study revealed the surprising results that binary embeddings maintain or even improve accuracy as model size grows, though their behavior is still being investigated. Finally, the readiness of approximate neighbor search for billion-scale protein databases highlights both the promise of current approaches and the opportunity to build more powerful biological foundation models. If we zoom out a bit further and reconsider the foundation model paradigm, we see that all these tools rely on common algorithmic and computational frameworks. The components presented here for scalable embedding inference and search are straightforward to adapt to other domains. In fact, much of the infrastructure here was adapted from a previous project of ours named HyperRAG, where we showed linear strong scaling out to 1,000 accelerators across both Polaris and Sunspot using Parsl. As progress continues to be made on FMs and scalable search, Parsl can help to connect big data and FM inference across a variety of domains with HPC resources to deliver new scientific insights. So thank you for listening to this talk. Much of this project spun out of our genome scale language modeling efforts, so if you have any questions or are interested in collaborating, please reach out to me via email. Lastly, we acknowledge the support from ALCF, KBase, and the CEPI Foundation, as well as special thanks to John Gerald at UIUC, who provided many insights on the applications we are pursuing.